Mm-hmm. Okay, so suggesting the SNVs, for these three brain disorders that have case versus control, the called SNVs are indeed enriched for brain-specific splice alteration. And then we take the two extremes. Remember our comparison to the expression mechanism. So again, we want to separate the splicing mechanism to expression mechanism. So we take on the left, two case examples. We take the genes that are only remember, all these genes here are already brain-specifically splice-altered, called through our SpliceTransformer. So then we take those that are only brain-specifically expressed, and then on the right are those universally expressed as comparison of two genes. And we can also compare all the genes, right? So on the left here, we plot all the genes that are called through SpliceTransformer, and the distribution, surprisingly, is a bimodal distribution, suggesting enrichment on both ends of these two cases. And then we take the two extreme ends and run through the Go enrichment analysis, and we identify this biological functions of the groups of genes. So for those genes that are brain-specifically expressed and brain-specifically splice-altered, these are related to neuronal functions like synapse signaling. Whereas for those genes that are universally expressed but brain-specifically splice-altered, those are enriched for actin filament and extracellular matrix organization, these type of functions. Of course, our method identified some novel genes that has never been reported. Those are this white background. So some of the genes have been reported, some are not. And we try to better understand the relationship of these three different brain disorders through a network view. And we were able to find that these genes share the same biological pathways, but within each pathway, the associated genes are different across the three brain disorders. And we further take a cohort of 95 patients with diabetic nephropathy, where we have both whole exome sequencing and renal tubule RNA sequencing data as validation set to further experimentally validate our method. And our validation rate was 83%. Here are some examples. I don't have time. And we made this publicly available with a browser web server. And just one last sentence, we have, based on the SpliceTransformer, we try to do a further benchmark on all the pre-trained genomic language models for a different variety of RNA predictive applications. And we were surprisingly found that for the large language models, it's not always the larger the better, but we found that the models that fit the biological context or the biological task better can make the best performance across all the benchmarks. And this is our team. We are located in the beautiful city of Hangzhou, and we are a young group, and we are hiring. Thank you.
Thank you. Time for one question while Dennis, you can come and set up your talk. Yes. So the classification of genes associated with disease, which you put at the end, was that based on variants or just on the prediction from reference sequence? These big clusters that you looked at.
The cluster of which one?
Network of genes.
Network of genes? Oh, those are the genes that are identified through SpliceTransformer that are brain-specifically splice-altered genes. Yeah, all the genes related to brain-specific splicing.
They differ from what we measure directly in GTEx? Or?
No, through our prediction. Yeah.
Okay, yeah. Thank you.